Okay, let's make a start while people are still joining. Welcome, everyone. My name is Judy Dansures Cabrera. I work for Roche. I will be chairing the webinar, and it is my pleasure to introduce today the webinar number 6 from the series on the Estimands Academy for Trial Teams, which has the title Estimands in Clinical Pharmacology with a Bioequivalence Case Study. The webinar is going to be recorded, and you will be able to find it in the PSI Video On Demand library in the FPI YouTube channel later. The speakers today are Mark Baird and Sue McKendrick from PPD, both of them, Heli Lingard from Novo Nordisk, and David Wright from AstraZeneca. With that, I'll hand over to Mark.
Thank you very much, Judith. So, hi, everybody. I'd first like to introduce myself. My name's Mark Baird. I'm a Director of Clinical Pharmacology at PPD, which is part of Thermo Fisher. I've been a clinical pharmacologist or a pharmacokineticist for over 20 years, actually pushing 25 years now. And today's webinar is based on a paper which I've co-authored with Heli and Sue, who are also presenting today. And we also have David, who will join a panel at the end of today's webinar. And I would also really like to recognize Essim, Vivian, and Florian, who are listed here as well, who really contributed hugely to the paper, but are unable to join us today. So the paper and today's webinar are a joint effort between clinical pharmacologists and statisticians. And I would say the target audience of today's webinar is primarily clinical pharmacologists or medical writers or clinicians, anyone really who's supporting clinical pharmacology trials. And we're really looking to recognize the benefits of using the Estimand framework in clinical pharmacology trials, as the title suggests, which is applying the Estimand framework to clinical pharmacology trials with a case study in bioequivalence. So today's webinar will be split into a few different sections with different people presenting. Firstly, with my clinical pharmacology background, I would like to provide a little bit of motivation and insight into why we're really looking to push this issue today. And then I'll hand over to Heli and Sue to introduce Estimands and the thinking process in a little bit more detail. And Sue and Heli will also present a case study where we look at a bioequivalence trial before I provide some brief summary and conclusions, and then we'll hand over to David and the panel and myself to answer some questions and comments. So if you do have any questions or comments as we go through the webinar, please feel free to post those, and Judith will collate those for us as we go through. So the motivation, before I jump into this in too much detail, I've got a couple of slides where I want to introduce two concepts here, which we're really focusing on. The first one is what are Estimands and what is the Estimand framework? So in the context of today's discussion, we're referring to ICH E9 revision 1, which was released in 2019, which introduces the Estimand as a precise description of the treatment effect, reflecting the clinical question posed by the trial objective. And the Estimand framework is essentially a framework to align planning, design, conduct, analysis, and interpretation of clinical trials. And it's aimed at everyone on the trial team. It's not a statistical topic, and that's one of the key points that I really want to get across today. Is it's for clinical scientists, physicians, sponsors, medical writers, statisticians, clinical pharmacologists, the entire team, and it's intended to provide clarity around the trial. So the objectives, the Estimands, and the design, and ultimately enhance and streamline interactions with regulators who are interested in and asking for the Estimand. And the other side of today's webinar is what are clinical pharmacology trials? What do we mean whenever we speak about clinical pharmacology trials? And simply speaking, we're really referencing all early phase healthy volunteer trials. Those can be exploratory trials, like first-in-man trials, such as single ascending dose, multiple ascending dose. But I would say we're more so focusing on the confirmatory clinical pharmacology trials, such as bioequivalence trials, drug-drug interaction, food effect, renal hepatic impairment, lactation studies, and concentration QTC analyses. Really, any study where we're looking to confirm statistically an effect on the pharmacokinetics of a drug. So why are estimands important? The ICH guidance, which was released in 2019, states that central questions for drug development and licensing are to establish the existence and to estimate the magnitude of treatment effects. To properly inform decision-making by pharmaceutical companies, regulators, patients, physicians, and other stakeholders, clear descriptions of the benefits and risks of a treatment for a given medical condition should be made available. Without such clarity, there is a concern that the reported treatment effect will be misunderstood. And estimands are also important to strengthen dialogue between disciplines, as well as the dialogue between the sponsor and the regulator. And when do we apply estimands? Again, looking at the ICH guidance, it states that estimands are relevant whenever a treatment effect is estimated or a hypothesis is related to a treatment effect is tested, and regulatory interest in the application of the principles outlined in the Estimand Framework will be greater for confirmatory clinical trials, where used to generate confirmatory conclusions for data integrated across trials. But our viewpoint essentially is the Estimand Framework is relevant to all trials. So why use estimands in clinical pharmacology trials? What's really driving us to push this as well? So in 2022, which is approximately 3 years after the ICH Estimand guidance was released in 2019, the FDA updated their statistical approaches to establishing bioequivalence guidance for industry to actually incorporate Estimand terminology. Specifically, lines 324 to 335 of this guidance state that the trial protocol of a bioequivalence study should include the following components of an estimand, and it goes on to list 5 separate components of an estimand. So the FDA have already started to update their guidances to incorporate estimands, and I would also like to emphasize that 2 of our co-authors of our paper are also from regulatory agencies. Essim from the MHRA and Florian from the European EMEA. So regulatory interest in this topic is not unique to the FDA. Looking at the current climate, and this is very much a personal perspective based on the studies that I see and I'm involved with here in a CRO, there appears to be a very slow uptake of the use of estimands in clinical pharmacology trials. And why might that be? Is it due to a lack of awareness? Is it due to a lack of training? Is it perhaps the incorrect assumption that estimands are a statistical topic? Or maybe there's simply just resistance within the field where there's this mindset where we know what we're doing. And I would emphasize that I am coming from that background. I am not a statistician. The idea of estimands is not something that just naturally, was a good fit for me and my background. And we're looking at studies here which each clinical pharmacology study design or type has its own detailed guidance from the different regulatory agencies. And actually, there are product-specific guidances as well. So if you're running a bioequivalence study, you have your bioequivalence guidances, and you've also got product-specific guidances. So really, do we need another framework to follow? And other than a gentle introduction within the FDA bioequivalence statistics guidance, there's no real discernible push for estimands in clinical pharmacology trials. So there's a multitude of factors here that are probably leading to a slow uptake. And this is one of the things we're hoping to address with our paper and today's webinar. Again, from my personal perspective, as I say, I'm not a statistician, and Estimands were a tricky topic for me, to be honest, when I first got involved in this. But the main driver for me is I often see early phase clinical pharmacology trials coming through where the study design, the intention of the study is very vague, and you need to infer or guess what the study is doing. And a couple of examples here. One is a bioavailability or a bioequivalence example, where I often see objectives coming through in a protocol or a synopsis, such as to determine the pharmacokinetic profile of drug A following the administration of 2 different formulations of drug A to healthy volunteers. So straight away, I'm not sure, is this a bioequivalence trial? Is this a relative bioavailability trial? Do we expect the treatments to be different? Do we want them to be the same? Are we powering this statistically for a sample size to meet acceptance criteria, or is it more exploratory? And another good example are DDI studies, where often we'll see an objective which simply states to evaluate the drug-drug interaction potential between drug A and drug B. And again, I'm guessing which drug's the perpetrator, which is the victim. Do we need to analyze both compounds? How frequently do we have to analyze both compounds? And I can get from the top to the bottom of a protocol and not actually understand what the objective and the design of the study is intended to do. The Estimand framework is more robust and designed to tackle this. And another contributing factor, sorry, Helly, is the prospect of regulatory success. So Essim, who's one of our co-authors in our current paper, along with some of his colleagues, have released a paper in 2020, which dates back to the 120 assessment reports, which were submitted to the EMA between 2013 and 2018. Based on their review over that 5-year time frame, they reported that 22%, or 120 out of 551 submissions, included major objections in the clinical pharmacology section of the assessment report. So almost a quarter of those submissions had PK, clinical pharmacology related issues, which were associated with the target population, some PK-PD relationship issues, even bioavailability and bioequivalence issues. So we anticipate that perhaps applying the Estimand framework here might reduce these deficiencies related to clinical pharmacology and potentially increase the potential for success. So Estimands in clinical pharmacology. Applying the Estimand framework will ensure that the objectives, the endpoints, and assessments are clear and transparent. Different aspects of the trial are aligned to the question or the estimand, so the design, data collection, data analysis, and interpretation. The data issues are considered at the design stage, not towards the end of the study around database lock. Crucially, the treatment effect is not misunderstood, and ultimately will lead to more effective sponsor-regulatory interaction. So from my point of view, there really is a strong case here for using the Estimand framework across many, if not all, clinical pharmacology studies. So I'd like to just hand over to Helly now and Sue to provide a little bit more insight and detail around the Estimand framework itself.
Thank you very much, Mark. This figure illustrates the Estimand framework as laid out in the ICH E9 addendum. And it very clearly separates the question we want to ask in our trial from the description of how we intend to answer that question. The study objective and the estimand, they clarify our question, and it's not until this has been fully specified that we actually supposed to begin discussing how can we answer that question. So the order is important, and you should never begin your trial planning by discussing a specific design or a specific statistical methodology you would like to use in your trial and then go backwards and try to formulate a question. That is the wrong order. But it may be that in cases that you have fully specified your question and you initiate discussion of how to answer that question, that you do find out, for example, that you don't have any statistical methodology you can apply to help you answering your question. And in that case, you need to go back and revisit your question. So even though the order is important, it is possible to make iterations in the framework. So the estimand consists of 5 pieces of information, the so-called Estimand Attributes. The attributes are interrelated and should not be seen in isolation. And some of the attributes are already familiar to you. The description of the treatment condition or conditions you are including in your trial, the endpoint you're interested in, and how you want to summarize your treatment effect. The target population is the population that is targeted by the question you're asking in your trial. This attribute is a little bit complicated in a clinical pharmacology setting, or can be. And today we will not focus on this attribute, but we will refer you to our paper where we have included a short discussion of this specific attribute. Instead, we would like to focus on the last attribute, the intercurrent events, and the strategies we use to handle these events. So what are intercurrent events? Intercurrent events, according to the ICH guidance, is events occurring after treatment initiation that affect either the interpretation or the existence of the measurements associated with a clinical question of interest. Existence refers to the so-called terminal events, beyond which the measurements or the endpoint cannot exist. A common example of a terminal event is death. And then you could argue, well, most of my PK trials, they are not, we never see death in such trials. They are short, and very often they are conducted in healthy volunteers. And that's correct, but it may be also that you are running a trial in severely ill patients, or you have a trial with a prolonged exposure. And in that case, death can be an important intercurrent event to consider. The ICH guidance and the associated training material that you can find on the ICH website, they are both very much focused on clinical efficacy trials. So the examples of the intercurrent events in those documents, they are perhaps not that relevant in a clinical pharmacology setting or in a PK trial. So when you want to identify intercurrent events in your PK trial, what you should think of is what are the events that can impact the PK processes in the body of the participant? Or in other words, what are the events that can affect absorption, distribution, metabolism, and elimination? One example of an event that can impact absorption is a dosing error. Use of interacting drugs can impact distribution and metabolism, and formation of anti-drug antibodies can impact elimination. So it's things like that you need to consider when identifying intercurrent events in your clinical pharmacology trial. Missing data is not the same as intercurrent events. This is something separate from intercurrent events. Missing data are data that exist and are relevant for our statistical analysis, but unfortunately were not collected in the trial. So measurements that are not collected in the trial, they do not impact the PK processes in the body of the participants. So therefore, these are not intercurrent events. Very often, protocol or intercurrent events are confused with protocol deviations. They are not the same, but there is an overlap. So in this slide, we have provided a few examples of events that are both intercurrent events and protocol deviations. For example, violation of the fed and fasted condition, use of interacting drugs, treatment non-compliance, and dosing errors. Examples of events that are protocol deviations but not intercurrent events are sample collected outside of window or missing blood or urine sample. Neither of these two examples will affect the PK processes in the body of the participants. So therefore, these events are not intercurrent events. So examples of intercurrent events that are not protocol deviations are immunogenicity, pregnancy, again, maybe not that commonly seen in PK trials, treatment discontinuation due to tolerability issues, side effects such as vomiting and diarrhea, and death. So now it's time for a quick quiz question. We have made up 4 events, and I hope there will be a window popping up on your screen so you can make a vote. We kindly ask you to classify each of these 4 events as either an intercurrent event only, a protocol deviation only, both an intercurrent event and a protocol deviation, or neither of them. So the first event is blood sample taken outside of window. Second one, intake of food or drink not according to administration instructions. The third one, participant withdraws consent 6 hours after dosing. And the last one, formation of anti-drug antibodies. So I'll just give you a minute to cast your votes. Okay, I think we are almost there, ready to reveal the answers. So the first event, blood sample taken outside of window, as most of you have voted, this is a protocol deviation only. It will not affect the PK processes in the body of the participant. And the second one, again, the majority picked up that this was both an intercurrent event and a protocol deviation. The third one, participant withdraws consent 6 hours after dosing. That was a bit tricky, I can see. So it's actually neither a protocol deviation nor an intercurrent event. That a participant withdraws consent does not impact the PK processes in the body. And also, participants in clinical trials can always withdraw. That will not be a protocol deviation. So the last one, formation of anti-drug antibodies, as most of you voted, it's an intercurrent event only. So thank you for your engagement, and I'll hand over to Sue, who will take us through how we can handle these intercurrent events if they happen in our trial, and also to introduce the case study.
Thanks, Helly, and hello, everyone. So there are 5 strategies for handling intercurrent events that are described in ICH guidance. Other strategies may exist, and they can be used alone or in combination. So, for example, I might choose the treatment policy strategy for formation of anti-drug antibodies and the hypothetical strategy for use of interacting medications. Relevance depends on the context. For example, death could be handled by while alive strategy or by considering a treatment failure. Because considering it as a treatment failure in a composite variable strategy. But as we're going to see in the next slide, treatment policy strategy is impossible for death. So let's take this PK example. The treatment policy strategy, as you can see from the definition, it requires follow-up and collection of data following intercurrent events, because the value of the variable of interest is used regardless of whether or not the intercurrent event occurs. So you can see that is rather impossible if somebody's died. But anyway, as Heli mentioned, that's not the most common situation for PK studies. So let's think about a PK example in a hospital setting where an infusion should be given for 4 hours. And let's consider the intercurrent event of an incomplete infusion. So in the graph, we have one person with a green line who has a complete infusion and so no intercurrent event. Another person with the red line needed to be stopped at 2 hours due to an adverse reaction. So if we're interested in the treatment policy strategy, then we're interested in their areas under the curve, regardless of the actual infusion time. So that infusion stopping early or being interrupted is actually part of our treatment conditions of interest. For this strategy, we would consider it clinically relevant, and that much smaller AUC of the red curve would be incorporated and it would reduce our average estimate. So if we are trying to compare two different formulations, the impact of incomplete infusions would be incorporated into our treatment effect of interest. And perhaps that's not the most relevant strategy for our questions. Let's move on and let's talk about the hypothetical. So a hypothetical strategy would be interested in trying to remove the impact of the incomplete infusion so that the treatment effect of interest would be the difference in formulations alone. So here we would be interested in the treatment condition described as an infusion of drug over 4 hours as though the full infusion was administered without interruption, i.e. in the graph, the dotted red line. So there can be different possibilities for hypothetical scenarios we might wish to explore. And in estimation, there are different possible analytical approaches to infer the AUC value you might have observed on this individual had the full infusion been given. So some kind of imputation or prediction is required. Let's move on to think about the next strategy, which is the while on treatment strategy. So that's defined as the response to treatment prior to the occurrence of the intercurrent event is of interest. So for this strategy, we would focus on a different endpoint rather than AUC 0 to infinity. So we might focus on Cinf, which we're defining as the concentration at steady state just prior to infusion stopping. Or else we might calculate the AUC up to the end of infusion and divide by time on infusion to derive a measure of average exposure, which perhaps we'll call CAV. So note that there is no implied imputation or prediction with this strategy. We are focused on a different endpoint. Okay, we'll move on now to the composite variable strategy. This is another strategy that alters the choice of the endpoint. As per the definition, an intercurrent event is considered in itself to be informative about the patient's outcome and is therefore incorporated into the definition of the variable. So often with this strategy, it includes undesirable intercurrent events as treatment failure. So it's pretty popular in phase 3 efficacy, and I noticed that the FDA bioequivalence guidance strangely mentions it too. So clinical responder endpoints, however, are much less common, I think, in PK studies. But let's just imagine in a situation to give you a feel for what it is. So let's say we've got a therapeutic window and it's depicted on our graph by the area between the two horizontal dotted lines. So our green line person, they're a treatment success because they meet the therapeutic window and they received the complete infusion with no intercurrent events. Now, there are two different ways to be incorporated as a treatment failure in this example, which is of a binary, an example of composite variable endpoint. So we've added an extra person on the plot compared to the other pictures you saw. So firstly, our solid red line person who stopped the infusion early due to adverse reaction. So they are a treatment failure because of that stopping of the infusion, and they're depicted with the red cross. And then there's another individual with the dashed red line who fails to reach the concentrations within the therapeutic window. So they might not have the desired efficacy, and they would be also classed as a treatment failure. So as an aside, you can also imagine using a composite continuous endpoint where perhaps a value of 0 is taken for stopping infusion for tolerability issues. And that might then lead you to summarizing the CINT values with a median rather than a geometric mean, for example. So there's lots of possibilities, but I would suggest it's perhaps not the most common choice of strategy in PK. Okay, so let's move on to the principal stratum strategy. So the definition of principal stratum strategy is focused on the target population of interest. So that might be taken to be the principal stratum in which an intercurrent event would or would not occur. So let's imagine we're interested in the objective that, you know, sort of an objective like Mark was thinking about, where we're looking at relative bioavailability of a test to a reference. And that will keep thinking about the 4-hour IV infusion example. And we want to look in healthy adults who are able to tolerate the full infusion of both the test and the reference formulations. So i.e. stratum 1 in the table. So the principal stratum excludes strata 2 to 4 of those who would have an adverse reaction for one or both treatments, which would mean that the infusion would need to be stopped. So remember, we are forming our question as it relates to the target population. So, you know, having done that, we might then decide that we're going to design a crossover to look at this situation. But for a parallel group study, it is hard to define who would be in each strata. Okay, because we only know response to one of the formulations. So if we know someone has no ICE or intercurrent event on test, we know they must be in strata 1 or 2. If we know someone has no ICE on reference, we know they must be in strata 1 or 3. So there are issues with estimating this strategy using a per protocol set in a parallel group study, unless we can assume, or make what might be a big assumption, that no one would be in strata 2 or 3. So 2 key principles to think about. The point of Estimands is we're trying to define a treatment effect of interest by comparing treatments across the same population, the same target population. And then another important principle is that we should have preservation of the balance we have in randomization. So the ICH guidance, if you read it, you know, they are very much focused on parallel group efficacy studies, and they do give quite a lot of criticism to per protocol sets for that reason. Anyway, we'll move on. And let's have a look at this top-down process that Heli mentioned in a bit more detail. The training material that ICH released describes this structured and logical thinking process. Step 1 considers the therapeutic setting and intent of treatment determining the study objective. Step 2 identifies the intercurrent events. Step 3 discusses the strategies to address them. And when I say discusses, I'm thinking about that trial team discussing this together. Okay. Step 4 is to describe our treatment effect of interest, the Estimand. So that should have built our objective into a much more precise clinical question of interest of what it is we want to find out. And we try to establish that before we move on to how. And then we can think about step 5, aligning the choices of trial design, data collection, and the method of estimation. And step 6, to consider assumptions and sensitivity. Anyway, in this webinar, we are going to focus on kind of steps 1. Well, I guess we're going to focus on steps 1 to 6, actually, but mostly perhaps we were focusing on the what, perhaps. But anyway, resist. I guess one of the key points is resist temptation and don't be the kangaroo who jumps straight to step 5. We're also focused on data that we often think about that analysis and estimation steps first. Don't do that. Really get clear what is your what. Okay. So rather than skipping like the kangaroo, I'm saying glide like an eagle. So the Estimand eagle has sight of the clinical landscape, that big picture of what we're trying to achieve. But they also have that eye for detail and glides purposely to its target. So we are going to have a think about applying this Estimand thinking process and Estimand framework to a bioequivalence case study. It's been a long journey. I think we started thinking about this study in 2022, but yeah, it's a pharmacokinetic bioequivalence study comparing pirfenidone tablet and capsule dosage forms in healthy adult volunteers. So this paper came out in 2017 before the ICH guidance. And we did try contacting, emailing the contact author, but we didn't get any response. So failing that, David suggested, David and I are both Doctor Who fans, and we thought, well, maybe we can go back in time and get some kind of time machine. But obviously, yeah, that didn't work either. So this is our time machine disclaimer. That we couldn't get the pirfenidone trial team around the table, and we might be missing some important information. But hopefully this will be useful as an illustration of applying the Estimand thinking process to a sort of real situation. Okay, so next slide. So let's start with step 1. Thinking about the therapeutic setting, intent of treatment, and determining a trial objective. So first of all, what is pirfenidone? It's an oral anti-fibrotic agent, and it's used to treat serious condition of idiopathic pulmonary fibrosis. This drug has quite a few side effects around the GI tract, nausea and vomiting, which is much worse when fasted, and there's also a tolerability issue with rash. And in clinical practice, it's titrated up to 801 mg 3 times a day. So you can imagine taking 9 of these capsules a day. Okay, and the intent of the new formulation is it would just be one tablet 3 times a day rather than those 3 capsules. So it's a convenience, and we're hoping for similar tolerability and PK. So after our discussions, we think the objective is to evaluate the rate and extent of absorption of pirfenidone of a new high-dose tablet formulation of pirfenidone, with the goal to establish equivalence to 3 capsules in the fed state. And note, the original trial also studied the fasted state. Typical endpoints that we've got in mind are area under the curve, 0 to infinity, and Cmax after a single dose. And I am going to pass over to Helly to continue the journey through the thinking steps.
Yeah. And the next step to consider is what are the intercurrent events? And please bear Sue's disclaimer in mind. It may be that we have missed some important information, so we haven't identified all relevant intercurrent events, but at least we have identified some, so we can make the illustration. So the first one, vomiting or diarrhea impacting absorption of treatment, was actually implicitly identified when we had this thorough discussion about in step 1 that Sue just took us through. Because we learned that pirfenidone induces vomit and diarrhea in many patients. We would really like to distinguish between events that are related to pirfenidone and those who are not. And we would like to make this distinction because we would like to apply different strategies to the 2 situations. And be aware that if you do or make this distinction, you need to be able to justify, especially the events you classify as non-related to pirfenidone, in fact, are non-related. But in this case, we believe we can. The second intercurrent event does not take both formulations, is actually an example where we had to do an iteration in the Estimand Framework. So we didn't identify this intercurrent event in the first run or in the first cycle. It was not until we began discussing which trial design to use, and we agreed that a crossover would be relevant, that we realized that then we missed an intercurrent event, and we had to go back and reword our question. Again, for this one, we would like to distinguish between events that are related to pirfenidone and those that are not. The third intercurrent event is dosing deviation, and the last one is use of interacting drugs. Next step is to discuss the strategies for handling these intercurrent events. And as Sue just showed us, around 60% tolerates the highest dose of pirfenidone. So there's quite a fraction that does not tolerate this high dose. And the new tablet formulation is that high dose. So we believe it's only relevant to evaluate bioequivalence in those who can tolerate both formulations. The remaining intercurrent events we propose handling with a hypothetical strategy. So overall, in general, you could say that we are evaluating bioequivalence as though these events did not happen. So now we are ready to construct the estimand or to word our question. And we need, of course, to make sure that we address all the estimand attributes. An example of how we could word our question is the following. What is the geometric mean ratio of test versus reference of pirfenidone AUC 0 to infinity after a single oral dose, comparing test to treatment both administered with food as though dosed correctly with a standard meal, without intake of interacting drugs or substances, and without vomiting or diarrhea due to unrelated tolerability in healthy adults able to tolerate both formulations. Wow, a long and detailed question. And we will even have one for one separate one for Cmax, we will replace the endpoint AUC not to infinity with Cmax. But it's also very detailed and clear what we are trying to estimate in our trial. The red or the pink text in the treatment conditions attribute refer to the intercurrent events that we chose to handle with a hypothetical strategy. And the pink text in the target population attribute refers to the two intercurrent events we propose to handle with principal Stratum strategy. So now we have constructed our question or worded our question in detail. So now we're ready to discuss how can we answer this question. The first thing to do is to discuss the trial design. And as Sue explained, it seems not to be relevant to evaluate bioequivalence in the fasting state due to these tolerability issues. And of course, this is something we need to agree with regulators upfront before we initiate the trial. But we propose a 2x2 crossover in the fed state and of course with sufficient washout. Because we have identified the intercurrent events upfront, we would probably be able to set up some mitigation plans to minimize the extent of some of these intercurrent events. And this we can do because bioequivalence trials are conducted under very strict and controlled conditions. So by doing that, we would increase the quality of our trial conduct. Next, we need to define the analysis set we will be using when we estimate our geometric mean ratio. In the past, you'll be used to defining your analysis populations by looking at which participants would contribute with data in the statistical analysis. This is not sufficient anymore. You also have to specify which data points you will be using in relation to intercurrent events. And for PK trials, this means that you should consider this both on the concentration data level and the endpoint level. So in this slide, we have made up 6 examples of what could happen in our pirfenidone bioequivalence trial. And you can immediately see that participant 1 and participant 6, they experience pirfenidone related tolerability issues. And because we do not want to evaluate bioequivalence in those who cannot tolerate pirfenidone, we will exclude these 2 participants completely from our analysis set. That leaves us with 4 participants. Participant 2 and 3, they both experience dosing deviations. So we will remove the affected or the data from the affected periods, but we will keep, of course, the data belonging to the unaffected periods. Participant 4 uses prohibited medication in period 2. But as you can see, it is being used sometime after Cmax occurred. So for the evaluation of Cmax, we would be able to use data not only from period 1, but also from period 2. Whether or not we can use data from period 2 to derive an AUC as though prohibited medication was not taken, we don't know. That remains to be investigated. It will depend on exactly when this prohibited medication was taken. So participant 5 is all good, no intercurrent events, and we can use all the data from the 2 profiles. So these are the kind of considerations you have to make when you define your analysis set. Next, we need to decide which statistical methodology we should use to estimate the geometric mean ratio. And we propose a linear mixed model on the log scale, and we will use the standard bioequivalence limits for both AUC naught to infinity and Cmax, and use that to evaluate if we have demonstrated bioequivalence. So these were the 5 steps, the 5 first steps of the thinking process. So now we are ready to compare the 2 approaches, the original 2017 trial approach and the EAGLE trial approach. So for good reasons, they did not discuss intercurrent events and strategies for how to use them because, as Sue explained, this trial happened before we had the final ICH guidance. Whether or not they had some considerations along these lines is unclear. We don't know. We identified the intercurrent events up front and used that to set up mitigation plans to minimize the extent of some of these intercurrent events. And we also discussed and agreed on the strategies to use. In the original trial approach, they compared test and reference when administered both with food, but also fasted. As explained, we only suggest to compare the 2 formulations when administered with food. This led also to a difference in the proposed trial design. The original one was a 4-period crossover, and we proposed a 2-period crossover. In the original approach, they used 2 separate fixed effects ANOVAs, one for fed and one for fasted, and they used the so-called completers data set. We proposed a mixed model on those who can tolerate pirfenidone, and by that, we can also use more data than just the data from the completers. Unfortunately, in the original trial, they just missed the BE limits for Cmax. And of course, you would expect that our estimated in the EAGLE trial geometric mean ratio estimates would be different. And you could speculate that because we're able to use more data than the original approach, that we would also have an improved precision. And it may be that we were then able to meet the BE limits for Cmax. We of course don't know, but we can speculate. So with that, I will hand back to Mark to give the concluding remarks before we open the floor for questions.
Thanks, Ellie. So just to conclude briefly then, regulators place the emphasis of the ICH E9R1 Estimand Framework guidance on questions which impact drug labeling, and that includes confirmatory clinical pharmacology trials such as bioequivalence, DDI studies, food effect, etc. The thinking process itself adds value to all trials, clarifies trial objectives, and may impact the proposed design. And much like Sue said earlier, let's not be kangaroos and jump ahead. Let's glide like an eagle and identify what you want to find out before how. So just before we pass back to Judith and open the floor for questions, I just want to highlight some references here that we've mentioned throughout the webinar today. And also we have some acknowledgements as well, which we'd like to share on screen, specifically again to emphasize Essam, Kerwash, Vivian Lanius, Florian Lash, and also David, who joins us now as well as co-authors of the paper. So Judith, back to you.